Next, we're going to talk in a little bit more detail about the different kinds of viruses that infect different kinds of cells. So let's start out with bacteriophages. Again, bacteriophages, this is a really long name, so scientists oftentimes want to make things easy, so they'll simply just call them phages. These are going to be viruses that only infect bacterial cells. And again, as I mentioned in the previous video, it's interesting that the simplest of cells get infected by some of the most complex viruses. The bacteriophage virus kind of looks like that lunar space landing module. So you've got the head. In the head, usually you're going to have the DNA genome that bacteriophages have. And then you've got this little tail piece here, and then you've got these little tail fibers. And what happens is this bacteriophage will land on the surface of a bacterial cell, and it'll grab onto it, and then it'll use that little tail piece to actually inject the DNA from the head through the tail piece into the inside of the bacterial cell. So really it's only the DNA that gets into the bacterial cell, and the capsid just kind of floats off as space trash, essentially, outside of the cell. And so then once the DNA gets into the inside of the bacterial cell, then it can take that bacterial cell hostage and make more of these bacteriophage particles. All right, animal viruses. These guys are very diverse, and there are typically two key variables that are used to classify animal viruses into different categories. So first of all, we determine whether the genome happens to be made out of RNA or made out of DNA. And then we classify them based on whether the nucleic acids are single-stranded or double-stranded. And honestly, animal viruses can have every permutation of these two possibilities for each of these two categories. So for example, they can have a single-stranded RNA genome, double-stranded RNA genome, single-stranded DNA genome, or double-stranded DNA genome. So again, very, very diverse. On top of that, animal viruses also frequently tend to have viral envelopes. And this is going to help with infection of the host cells. Think about it this way. Not many other viruses that infect different kinds of cells, so for example, not bacteriophages and not plant viruses, neither of those will have viral envelopes. But animal viruses do. What's different about plant, bacterial, and animal cells? Well, plant cells and bacterial cells have cell walls, right? These rigid structures on the outside of the cell that helps to protect the cell. Animal cells do not have a cell wall. We just have a membrane. So it makes a lot of sense that the viruses that infect animal cells have these membranes, because if a membrane bumps into a membrane, they are highly fluid and they will just sort of mesh together, and it's a very easy way for an animal virus to get into the actual animal cell, so it can go through the replication of the host particles. So again, just to sort of compare, contrast animal viruses versus bacteriophages, typically bacteriophages have DNA genomes, whereas animal viruses can have the whole gamut of different kinds of genomes. And then bacteriophages, very few of them, if any, have an envelope, and very frequently animal viruses will have that viral envelope. So let's talk a little bit more about this viral envelope. Again, many viruses that infect animals have this membranous envelope. And the envelope also will typically have those glycoproteins attached to it, right? The pins in the pin cushion. And the viral glycoproteins are really there to help interact with the host cell, find the right cells to infect so that the host cell can so that the virus can actually get into the host cell and cause infection. When we look at the viral envelopes, typically viral envelopes are derived from the host cell's membrane itself. And so what we're going to do is we're going to take a look here at an example of how an animal virus will go through its replicative cycle. So in this case, this big thing right here is the host cell. This poor host cell is going to be infected with this little virus over here. So this virus has a single-stranded RNA genome. It has a spherical capsid, and the whole thing is then enveloped with this viral envelope in yellow, and that viral envelope is again studded with those glycoproteins. The virus will bump into this host cell, and those glycoproteins will interact with proteins on the surface of the host cell. It'll say, yes, this is a match, I can infect this cell, and then the membranes will fuse, and the capsid and the viral genome will get into the inside of the host cell. Again, this will fall apart. That will reveal the genome to the cell, and the cell will take over, essentially expressing and copying that viral genome. In this case, this is an RNA viral genome. So the cell will use this to make a template for making the mRNA. mRNAs are used to make proteins. There are two different kinds of proteins that need to be made for this particular animal virus, right? You've got to make the capsid proteins. These are typically going to be made using the free ribosomes inside the cell's cytoplasm. And then you also need to make these viral glycoproteins that are going to be stuck to the outside of the viral particle. Here's where the endomembrane system comes into play. And so an organelle called the endoplasmic reticulum will help to make, along with the bound ribosomes here, will help to make the viral glycoproteins. It'll be packaged by the Golgi, shipped to the plasma membrane, and those viral glycoproteins will be shoved to the outside of the plasma membrane, and they'll just sit there and wait until the rest of the viral particles are put together. Speaking of the rest of the viral particles, right, we've already made the capsid proteins using the mRNA, but the cell is also going to copy this template RNA to make more copies of the genome for those viral particles. Once you have the genome copies, the capsid proteins, these two parts of the particle will self-assemble, and then the whole particle will essentially bud off of the host cell. So the host cell remains alive, and it just keeps pumping out these viral particles. And as the viral particle leaves the host cell, it'll take the host cell's plasma membrane with it that has all of these viral glycoproteins stuck to the outside. So here's your new little virus that has just budded off this host cell. It can go on and infect neighboring healthy cells next door. Okay, so we kind of understand that, yes, there are viruses that infect animals. We understand the basics of how an animal virus gets into an animal cell and goes through replication. What we haven't really talked about yet was how do viruses actually really make us sick? So there's a variety of different ways through which this can happen. So typically viruses are either going to damage or they're going to kill cells. And they can do this by, number one, either causing the release of hydrolytic enzymes from the lysosomes, right? So we know animal cells have the lysosome. This is the stomach of the cell. And when you break open the lysosomes, that's essentially going to chew every part of the cell from the inside out until the cell is gone. Number two, viruses can also cause the infected cell to produce toxins that can lead to disease symptoms. These toxins, the cell doesn't know it's making them, but it makes them anyway, and it causes cells in the organism to become sick. And then number three, viruses themselves can actually contain these molecular components like those glycoproteins that are stuck to the outside of the envelope that can actually be toxic. And when they come into contact with a host cell, it can make that host cell sick. So what do we do to protect ourselves? Our best protection against viruses is preventative, typically vaccines. So a vaccine, when you go to the doctor and you get a vaccine for the flu or measles, what you're doing is you're allowing the doctor to give you a harmless derivative of that virus. It might be the virus itself, but in an incapacitated fashion. So the virus is essentially unable to cause infection, or it might not even be the whole virus. It might just be part of the virus, like one of those glycoproteins that are stuck to the outside. And when you give the vaccine to the system, what the system does is it starts to learn. You're teaching your immune system. You're saying, when you see this, either the virus or the glycoprotein or whatever part of the vaccine that it is, when you see this, recognize it as being bad. So that if I ever come across this virus in the real world that can cause infection, you as my immune system, you know exactly what to do, and you can eliminate it from my body before it actually makes me sick. So we're training our immune systems to recognize this bad virus. The vaccines, unless you have an allergic reaction to it, vaccines will never give you the actual disease that it's meant to help your body defend your body against. So, there are myths about, oh, I went to the doctor, I got the flu vaccine, and then I got the flu a couple days later. It wasn't because of the vaccine. It takes some time to build up the immunity against the disease that you got the vaccine for, and you may have just unfortunately got the flu virus before your immunity was built up. Or viruses also have the ability to mutate, some faster than others. The flu virus mutates very, very fast. This is why every year the flu is a little bit different and you have to get another flu vaccine to help combat the flu as it is currently progressing through its, mutations. When you go to the doctor and you're sick, right? And if your doctor does maybe a flu test, or your doctor suspects that you have a viral illness. Hopefully your doctor is a good doctor, and no matter how much you try to persuade them, or hopefully you don't try to persuade them at all because you've paid attention to these lectures, you don't ask for antibiotics, and they're not going to prescribe antibiotics to you, because for a viral infection, antibiotics are going to do absolutely nothing. What we've talked about, the amount that we've talked about antibiotics in this course, we've mentioned that antibiotics are usually drugs that target special things that bacterial cells do, right? Either they target the cell wall of the bacterial cell, or they will target the ribosomes of the bacterial cells and damage them and then cause the bacterial cell to die. If you were to take an antibiotic that targets the bacterial cell wall when you have a viral infection, viruses don't have a cell wall. So nothing's going to happen to them when you take this antibiotic. Viruses also don't have ribosomes. So if you take an antibiotic that targets bacterial ribosomes, again, the virus is going to be completely unharmed. So viral infections cannot be treated using antibiotics. Now, there are certain antiviral drugs that have been developed for certain viral diseases, like Tamiflu, right? You go to the doctor, they decide that they test you for the flu virus, and they say, here's some Tamiflu, go home, take it, you'll feel better in a couple of days. It's not a cure. All that the antiviral drugs really do is they kind of help to slow the process of viral replication and give your body a chance to catch up to eliminate the virus from your body. All right, now we've talked about bacterial viruses, animal viruses. Let's talk a little bit about plant viruses. So yes, even plants are susceptible to viruses. There are about 2,000 or more viral diseases that have been detected in plants. And typically you can tell when a plant is diseased if you see spots on the leaves, or they have a funky shape, or the fruits don't look right, stunted growth, damage to flowers, roots, etc. Now, most viruses that infect plants actually have RNA genomes. And you might be thinking, well, okay, I understand viruses can infect plants, but how does a virus get transmitted from one plant to the next, right? One plant is going to lean over and cough on somebody else, right? So there are two ways that plant viruses can actually be spread. There can be horizontal transmission. So this means there's a bad storm, right? We have these in Texas all the time in springtime, and branches break, right? So if you have one plant that is healthy next to another plant that is unhealthy, and you have branches that break on both of those, and the exposed tissue touches, you can transfer the virus from one plant to another. You can also have vertical transmission. This means that a diseased plant will produce seeds. Seeds are baby plants. And so the virus can get into the seeds, and when the seeds germinate, grow, sprout, the virus can then be in that seed and can contaminate or infect the new seedling that results from that infected seed.